This poster shown at the ESAC 2026 presents a comparative study of rAAV9 production using 3 different single-use bioreactor platforms, which is the orbital shaking, a rocking bioreactor, and a stirred tank, all using our proprietary EpyQ split 2 plasmid system. As we have a lot of experience on platform-dependent performance differences regarding the impact on a respective seed train and the later transfection efficiency and productivity, the aim of the present study is to compare these platforms regarding their potential for robust and high-quality rAAV manufacturing. In previous studies, the shaking platform has shown a slight superior performance regarding cell growth and cell quality compared to the rocking platform, but a strong improvement compared to the stirred tank bioreactor. As we then knew about the impact of bioreactor type on growth and quality of the cells during the seed train phase, we were wondering about the transfectability and productivity directly in those systems. With our standard animal-derived component-free suspension process, we consistently achieve titers of more than 1E11 Vg per ml across multiple serotypes and scales from 250 milliliter up to 200 liter in stirred tank bioreactors. For this study, however, 3 types of bioreactor systems were compared at the same working volume of 10 liter. The SB10X single-use bioreactor for orbital shaking, the ready-to-process WAVE 25 rocker for rocking motion, and the Biostat B 10-liter single-use bioreactor as a stirred tank. For all platforms, we used our proprietary HEC293 cell line, rAAV9 as the serotype, and the previously mentioned EpyQ plasmid system. Process monitoring for this study included cell growth, viability, nutrient and metabolic parameters such as glucose, lactate, glutamine, and ammonium, as well as analytical endpoints such as vector genome titer, capsid yield full to empty ratio, DNA impurities, and potency. The results show that other than for growth and cell quality, the rocking and stirred tank bioreactors are superior to the shaking system regarding the observed productivity. Both delivered better yields and higher product quality. A particularly interesting observation is that the shaking system continued to grow the cells for longer after transfection, which could be an indication for reduced transfection efficacy at otherwise comparable conditions to the other bioreactor systems. Overall, the metabolite trends were comparable across the platforms, indicating that the basic cell culture performance was similarly controllable. For crude lysate titers, rocking and stirred tank achieved significantly higher vector genome yields and capsid yields than orbital shaking. The full empty ratio also favors rocking and stirred tank. The impurity profiles include impurities derived from the capsid gene, the kanamycin resistance gene, and host cell DNA, which showed a strong performance of the rocking system on a, in general, relatively low level. In addition, the potency data showed that the generated vectors remain biologically functional for all 3 systems. The conclusion is that the rocking bioreactor delivered strong production and quality data, but is limited in its scalability. Despite the slightly higher plasmid-derived impurity we saw, the stirred single-use tank therefore remains to be our preferred platform for robust, large-scale rAAV manufacturing. The shaking bioreactor production performance suggests comparable to a well-developed seed train phase that for an efficient rAAV production process, a fast mixing time or increased shear stress seems to be required during either transfection or production phase. It also confirmed previous small-scale shaker flask production experience and therefore has proven its general upscale potential at the cost of lower yield compared to the other 2 systems using our stirred tank optimized process conditions. Continued growth after transfection in the shaking bioreactor suggests further potential transfection condition optimization by altering transfection or production phase mixing intensity. Careful investigation in the use of platform dependent on the process phase in question is, as we saw, required for a successful production. Seed train, growth, transfection, or production phase therefore need to be considered individually and evaluated in a final holistic approach to meet the production demand. If you are interested in further information, please write us at business@ascend-adv.com.